Uh, Bapi Raju from IIIT Hyderabad. I have the pleasure and I'm happy to introduce Professor Deva Priyakumar, my colleague at IIIT Hyderabad. I thank him first of all for accepting to give this invited talk today. Deva is currently a professor at IIIT Hyderabad, where he heads two entities. One is the Center for Computational Natural Sciences and Bioinformatics. And then he's also the academic head of the Technology Innovation Hub called iHub Data, which is a DST ICPS mission project, 5-year project that's running. He's the academic head of that. And his research interests are using computational chemistry tools to investigate chemical and biological systems and processes, and applications of modern AI and ML techniques for drug design, healthcare, and these applications in these domains. He has been the recipient of awards such as Chemical Research Society of India Medal, Indian National Science Academy Young Scientist Medal, Japanese Science Foundation and JSPS Invitation Fellowship, and Innovative Young Biotechnology Award. I'm very happy that he's giving this keynote talk on role of AI in the COVID-19 pandemic. Right, thank you. I hand over to Professor Deva for the invited talk.
Yeah, thank you, Professor Bapi, for a generous introduction. So, again, I really want to thank all the organizers and the program chairs for, kind invitation to be part of this, conference. So I will quickly share my screen. Can you see my screen?
Yes, sir.
Okay, thank you. So, as Professor Bapi said, I'm actually, trained as a chemist, but in the last few years, we have been, trying to use modern machine learning methods for, molecular design and also in problems in for problems in healthcare, right? So, we've been working with Professor Bapi and several others, and then trying to contribute to this particular area. So today, what I will do is, I will kind of, give a overview of how AI has possibly played a part in the COVID-19 pandemic, and then I will take, some examples of the work that we have done at IIIT Hyderabad. Okay, so this is the plan that I wanted to follow today. So I will spend the initial 10 minutes or so in trying to capture the larger picture of how AI is actually playing a role in healthcare. Basically, I'll talk about two things. One is disease diagnosis, which would be possibly the easiest to understand and then move further. And second one is, what are the ways in which, it can possibly make an impact, right? So that's the multimodal, how do we possibly use multimodal health data and make wiser and efficient decisions. So, and then in the later part of the talk, I will concentrate more on the role of AI in the pandemic. We'll talk about diagnosis. We did not do much work in the diagnosis part, but I will cover some work which have been, done by others, and then in prognosis, and finally the drug development work that has been going on. As I said, I will try to take some of the examples that we have been working on in the last 1.5 years or so. Okay, so to start with, right? Why AI for healthcare, or in what ways AI, seems to be making inroads into healthcare, right? So, these are some things that all of us would have heard at some point, where, you have these AI-assisted robotic surgery. There are some in the market which are already used, and then virtual nursing assistants, especially in case of, highly infectious patients, and then, elderly care and so on. And then the third aspect, which people have made significant contributions in the last decade or so, is in the medical diagnosis and in the medical image analysis. And finally, especially in the last 5 years or so, people have shown several examples where AI can be used for accelerating the drug discovery processes, which I will touch upon a little later. So these are some of the activities that has direct connection to healthcare, right? In terms of the patient care and diagnosis and treatment, so on. So on top of this, there is also admin around this healthcare, right? For example, insurance, or the managing the data, or the workflow, and then when it comes to drug discovery, the clinical trials, where we try to assess the efficacy and safety of a given drug candidate, to human population and so on, right? So there are several other areas where AI is playing a part. So today, for example, if we submit for like the medical insurance, right? The AI systems are shown to be extremely efficient in detecting frauds, for example, right? And so on. There are several other ways. But what is the consequence, or what do we want at the end, right? So, why, is it possible that the role of AI in each one of these and much more of the sub areas in medical care and healthcare, can this make the whole healthcare system more efficient? And then we are able to take healthcare to everyone, right? So especially in a country like India, right? The access of quality healthcare to everyone in the country is not possible today, right? But then, is it possible that this particular, technology can, make the system more efficient and, make a lot of these processes automated such that, we can cover more people under this, right? Okay. Basically, the question is, can we achieve, quality healthcare at a population scale? Right. So now what I will do in the next few slides is to just give some examples of how AI has been playing a role in diagnosis. I'll just take a few examples. As I said, so much work is going on in this particular area in the last decade or so. I'll take 2 or 3 examples from our own work. So this is a work that Professor Bapi and our group worked on it recently, right? This problem is simple. Now that you have a ECG signal, right, is it possible to detect any cardiovascular conditions based on this particular signal, right? This is again a problem that people have been working for a long time. Recently, we showed that you could take the ECG signal and then take the signals from different channels and beat level signal and things like that, and you can actually identify possible cardiovascular conditions very, very efficiently. And this accuracy reaches very close to the human expertise, right? This is, as I said, this is just one example. The second one example is, a similar kind of, signal where, we use what is called a gait, right? So, when people get older and if they are suspected to have Parkinson's, right? So one of the diagnosis is to have these force sensors on the feet, and then they let the patients walk and then measure this force on each of these sensors, and based on the force profiles, you try to identify whether this person is affected by Parkinson's or not, right? So here the signals, something the leftmost plot is the typical signals that you get when a patient walks, and then we were able to show that we could in fact come up with intelligent systems that can reasonably accurately help us in the diagnosis, right? And then the third, I'm just showing examples. As I said, one, the ECG signal. Second is that the force profiles from the measurements for Parkinson's diagnosis. And then third is, again, a different kind of signal. In this case, it's an image, right? Like the histopathology image or fMRI or X-rays and things like that, right? So this is a work, again, done by one of our colleagues in at IIIT Hyderabad in our group. And they basically were able to classify or diagnose renal cell carcinoma, right? It's a cancer diagnostics where you take the histopathology images, right? And then you're able to detect whether there are any cancer cells present or not, and then segment the patches where these lesions are present. Not only that, you could also sub-classify, or you classify based on the subtypes of different kinds of cancer. This is again another area of research where a number of our colleagues at IIIT are working on. Right. So let's go back and then talk a little bit more about the general impact of AI in healthcare. Right. So as I said, I started by giving you the higher level picture of what are the larger areas in which AI is playing a part, and then I just showed some examples where you take certain signal or a certain image and then try to do some diagnosis. Right. So the next step, what would be the next step, right? So the next step would be to see if we can actually combine different kinds of data, right? And then, not only diagnosis, can we have different kinds of learnings from this data, right? So, the slide that you see on the screen gives a larger picture of how data from multiple levels, like the genome sequences or the RNA profiles, all the way up to protein structures, and how you could possibly use them to learn multiple things in terms of the risk stratification or diagnosis of modern diseases like cancer, or understanding gene variations and possible risks for the future based on the genomics, and then the drug development and so on, right? So let's make the picture a slightly less complicated. So today, let us say, right, so when we talk about healthcare, right, so what are the things that we do? So one, of course, is that we try to improve our lifestyle in terms of diet or physical activities, right? And the second thing that we possibly do, or do an annual health check. And the third thing that we certainly do, or if we have some symptoms or signs, or if we have some difficulties with respect to pain, breathing, or any kind of difficulties, we go and get ourselves checked with the physician and possibly take certain measurements, whether it's a blood test or a X-ray or a CT scan or MRI. It could be anything, right? So that's the third level of healthcare that we seek for. And then the fourth is which is coming up these days is the multi-omics, where today people talk about Delta variant versus Omicron variant, right? So we could quickly check all those things today just by doing a simple genomic study. And then the fifth one, which is much more common these days, is wearables. Like we all of us have a Fitbit or a Mi watch or an Apple Watch or something like that, which constantly monitors our certain parameters, right? Like the heartbeat or the blood oxygen levels or the ECG, and things like that, right? So you have data being collected at multiple levels, right? It could be images, it could be temporal data like your ECG or heartbeats, continuously measured or often measured, and then specific tests, and then all the way up to multi-omics. Right. So today we do all these things for different reasons, and most of the times we do not look at all these data at a at the holistic level to understand the status or the health status. Right. So the next big thing that's happening in healthcare, or the possible, potential directions for higher impact of how AI can play a role in healthcare, is to be able to combine different kinds of data and then be able to do risk profiling, or early detection of diseases, or prognosis of diseases, and tasks such as those, right? So what would that mean? How can we possibly improve it? We are talking about different kinds of sensors which are monitoring our vital parameters, not just our heartbeat, not just ECG, but a different number of sensors that could possibly be used, that constantly monitor the health of different parts of the body, different organs of the body, and things like that, right? This is possibly going to be upcoming in the future, where we try to have wearables which monitor different aspects of health, and then this data could possibly be used for the things that I mentioned, like the early detection of diseases, diagnosis, prognosis, and then rehabilitation after a certain surgery or stroke, and things like that. Right. Okay. So now I hope what I tried to do so far in the first 15 minutes or so is to give a larger picture of what are the possible ways AI is actually playing a part, and then I take some examples of the traditional kind of things that we have been doing, where you take a certain condition, you get the biomarkers or images or anything corresponding to that, and then be able to predict that particular condition. And then we expanded our scope and saying possibly the future is in our ability to combine different kinds of data, and then at different intervals, different kinds of data, and things like that, to be able to cover a larger objective of better healthcare. Right. So now we go into try and discuss a little bit about how AI has played its role in the pandemic in the last 2 years. Right. So the four things that I will touch upon, or one is the diagnosis, second one is in the virology and pathogenesis, and third one is the drug development primarily, but then there is also a lot of work going on in vaccine development. And then the fourth one is in the epidemic transmission related areas. Okay, so let's talk about diagnosis. So this is something that we have not worked on, but a couple of our colleagues in the institute have been looking at the CT scans. Based on CT scans, how could we come up with AI-based solutions for diagnosis, right? So now that we are at a stage of pandemic where several of our either we own, or several of people who we know have been affected by COVID-19, and most of them recovered, but unfortunately a smaller population had to respond, right? So we also and every one of us, because of the situation, have been reading a lot about COVID-19, right? So one of the areas that people worked on much early is to be able to use the CT scans, right? So the images from the CT scans, and be able to detect COVID-19 and differentiate it from the regular pneumonia, for example, right? So this is a picture that I took from one of the publications. The references at the bottom, where you can see two different sets of images, one from a normal patient, and another from a COVID-19 affected patients. So a large number of groups have been working on developing modern machine learning based methods to be able to diagnose just given the image as the input. But largely, how does it work? Basically, you take all of these CT images and do some kind of image segmentation, and identify regions that, just give me 1 second, sorry. Okay, so now I can see my point. I hope you can see my pointer also. So the first step is that, to be able to, do an image segmentation using different kinds of methods. As I said, there are several tens of studies that have been published in the last 1.5 years or so, where use some deep learning method to do image segmentation and identify regions of interest or regions of possible infection and inflammation and so on, right? So, and based on those regions of interest, you extract features out of this and use those features again to use different kinds of algorithms to be able to detect whether the patient is affected by COVID or not. And then the results have been very, very impressive, right? It's as good as a medical expert can get, right? Because every given diagnostic tool or every given diagnosis, including the RT-PCR that we do, has its own limitations and has some inaccuracy associated with that, right? So the methods that have been published in the last 1.5 years have been very, very efficient to accurately identify COVID-19 patients from the others based on the CT images. So similarly, just chest X-rays, right? So people also use chest X-rays to get some clues as to how much of the lung is affected by COVID-19 and so on. This is possibly one of the earliest works that started right after the pandemic, where we said just X-ray is possibly the easiest thing that we could do, right? I mean, we can do X-rays pretty easily these days with a lot of mobile centers also being available, right? So once you are able to get these X-rays, is it possible to diagnose COVID, right? So one of the earliest things that happened. So similarly here, right? So basically a similar kind of approach where you start from the chest X-rays and do image processing and then train using different kinds of models and be able to predict COVID-19 with respect to the other possible chest conditions like the pneumonia due to bacterial infection or viral pneumonia and other lung-related diseases. Again, this have been reasonably successful, but but in terms of the real-life applications, I have not seen a larger impact in this particular area. I just mentioned this because this is possibly the earliest attempt towards using AI for COVID-19, right? So, I try to give a couple examples where we use the imaging diagnosis from, like the CT scans and the X-rays and use AI for diagnosing COVID-19, right? There are other areas, for example, you could also implement, people have also implemented, for the RT-PCR and so on, AI for RT-PCR and so on, right? And then the second one we'll talk about, maybe spend 10 or so, 15 minutes, 10, 12 minutes on this particular aspect where, this is possibly the prognosis aspects of, COVID-19 and to be able to predict how the transmission is going to be and predict the outcome or outbreak and things like that. Okay, so this is these are this set of studies that I'm going to show in the next few slides, or the efforts that we have undertaken at IIIT Hyderabad in collaboration with several partners like Intel and then Max Hospitals, Gandhi Hospitals in Telangana, and then Grace Cancer Foundation, IGIB, and so on, right? So there are several partners with whom we worked with in this particular aspect. So initially, what we did was this: like when when the COVID-19 started in China, that was in March, April timeline of 2020. We had access to this particular data of patients from a hospital in Wuhan. Right. So what is the data? The data basically is the complete electronic health record of the COVID-19 positive patients in that hospital. right? So all patients, COVID-19 positive. We also knew the outcomes, right? So this is the distribution of the data. We had let us say about 500 or so patients for whom we knew the blood reports and then their vitals and then their age and different kinds of all the measurements that one would typically do in a hospital stay. And then we also had the data of whether the patients survived after the stay at the hospital or they did not survive. If you remember, the initial days of the pandemic, the mortality rate was very very high, right? So what we did initially was to take this data and to be and we tried to see if we could develop a mortality prediction model. So what do you mean by that? Like, let us say, for example, right, this is the days to outcome. What that means is we knew the vitals and the other blood parameters of patients of, let us say, whether they survived or not 2 weeks or more, or let's say 10 days or more of the exact same set of patients, right? So now, is it possible to take this data and then predict whether this patient is likely to survive or not 10 days from now or 2 weeks from now, right? So why is that important? It is important because if we are able to confidently say that, okay, this is the group of patients who are at high risk, right? Maybe, we can, the medical system could concentrate more on those patients and, basically, let us say, give home care to the less risky patients. And then increase the chances of survival of this set of patients, right? So we use a very simple neural network model and also the other like the XGBoost and random forest kind of models, and then we try to predict what classify this set of patients whether they would survive or not, right? So as you can see, we do reasonably well by using a shallow neural network with respect to different measures. And then we also show that, we are able to predict the risk of this patient, or how risk, or, what is the probability, or what is the chances that these patients will survive or not, very well in advance with reasonable accuracy, right? So one thing that we realized quickly, right? So, and then we said, okay, this is not going to work for all patients, because even very early in the pandemic, we realized that, the mortality rates in, for example, in China and Europe and the US, let's say India, right? We saw different mortality rates, right? And a given model is not going to work on all cohorts. So, and hence we started collaborating with multiple, parties, and then we try to see if we can develop risk stratification and mortality prediction models for, Indian COVID-19 patients. That's how, we started, this, the second phase of the project, right? So where we took the data from, Gandhi hospitals and then Max hospitals in Delhi, and then we started working on a similar problem, right? So one thing you could quickly see in this, those who are used to looking at confusion matrices, right? One thing you can quickly recognize is that the model fails miserably, right? So, when I take the model developed on the Wuhan patients and then use that model to predict the mortality or the risk of the Indian patients, it's so bad, right? So that's what we expect, because as I said, the mortality rates are very, very different from, with respect to different populations. And then if you also look at the data distribution, right? So, the distributions of some important blood parameters with respect to the patients who survived and, did not survive between the two populations, right? The Indian and the Chinese populations, they are very, very different, right? So here you can see that, almost all the patients who had a neutrophil percentage of more than 90%, right? None of them survived, or most of them did not survive, right? But when you take the Indian population, you see, this is more than 90. This is, if you look at the green distribution, you see that more and more people survived, though the blood parameters say otherwise, right? So, and then hence we kind of developed a, similar models, but now for both risk stratification and mortality prediction, right? Mortality prediction, that's straightforward. What do you mean by risk stratification? When a patient is, diagnosed with COVID-19, the risk basically is that, one class is like, this patient, will normally recover in, 10 days, 2 weeks, whatever. And then the high risk patients are the ones who needed supplemental oxygen, or they had to be put on ventilators and so on, right? So again, we do, based on the data that was available, we do reasonably well in terms of predicting both risk and mortality for the Indian cohort, right? Okay, so let's, I'm not going to take too much of your time on this part of the talk. I think I'll show only one slide, but that kind of will cover, the kind of things that people do, right? Maybe, before I go to the next slide, maybe I will say a few things. So today, let us say you have the COVID-19. Let's go back 2 years. Let us say March, March 2020. Right. So when people did a genome sequencing, they had access to the complete sequence of the virus, right? So at that point, we did not know what kind of mutations are going to come in the future, right? We had several mutations that came, and two prominent ones, as I mentioned earlier, are the Delta variant and now the Omicron variant. So let us say, right, is it possible, if let us say, we asked a question saying, is it possible to predict what mutations are going to happen in the future, right? Or is it possible, let us assume a new virus disease has come up and then I do not know where the origin is. Just based on the sequence, is it possible to find out what are the possible hosts, whether it came from bats or some canine or, some other, animal, or it came from, somewhere else? Is it possible to find out, what was the host of this particular virus, right? And that this, for example, are the two particular aspects. We also have done some work on predicting the, hosts based on the sequence in terms of what host this virus has come from using AI methods. And the other project that we started, and possibly, one of our faculty is leading right now, is to predict future mutations based on the historical data, right? All these, of course, are very important. And then the third aspect is, let us say that we have new mutations, there are 100 possible new mutations is something that we predict, let us say, right? So now what I would like to know is this new variant going to be more infectious, or is it going to be more dangerous than the current one or the past ones? Right. So today, after the Omicron has come, based on whatever data that we have seen, we say that, it's milder than the Delta, right? But, is it possible to predict these mutations in, in advance, and then be able to comment saying, is it higher in terms of risk or lower? Is it going to be, more infectious or less infectious, and so on? Okay, so I will just take, I'll connect the things that I just talked about in the, last 10 minutes. As I said, right, so we collected, patient data from two different hospitals. One, about 2,500 patients from hospital in Telangana, and about 500 patients from Delhi. Right. So what are the data that we had? All of the clinical data, and then the radiology data like the X-rays and CT. And for this particular cohort, we also collected the samples from the patients, and then did the sequencing, and we have the viral genome sequences for them. Right. So as I said, we were able to successfully develop risk stratification and mortality prediction models. And then the other thing that is ongoing is the disease progression that I don't have much to show, but that is something that we could do, and we are doing. And then the third aspect, as I said, was the the multimodal thing that I tried to explain initially, right? So if you look at this part of my slide, right? If you look at this part of the slide here, what are these data? Like, for example, age is one kind of data. and then vitals like the heartbeat, and then the SpO2 levels, and then symptoms, whether this person has congestion or fever or certain symptoms, and then the lab reports like the neutrophils percentage, HC reactive protein content, and different kinds of blood markers. And then comorbidities. What are comorbidities? Whether the patient has history of diabetes or hypertension, or any surgical procedures, or was this patient under chemotherapy. These are some of the comorbidities. And then the last one is medication. People do take regular medication for different conditions, again, like the thyroid, for example, or diabetes, for example, right? So these are the kind of data here. And then immediately we switch to images, right? We have data from images. And then this is another extreme where we are talking about a viral genome itself. It's a long sequence of letters, right? So is it possible to use all of these data and then be able to talk about or understand this situation better, right? So here, as you can see, we were able to associate certain mutations with respect to disease severity, right? The disease severity meaning, whether that, patients with that particular mutation had to go through a longer COVID, or, had to get oxygen and things like that. And then we go back and then associate this to at the structural level, right? Because, once you have the viral genome, we can talk about structure of the genome, right? So, if we can connect all this back to the disease severity and mortality, right? So this is another work that we did, and then it's published again. So those interested may look it up, right? So how much time do I have? Can someone?
Yes, sir, you have time till 11.
Okay. All right, so then the last, I will take about 10 minutes or 12 minutes, and then see if, we can leave some time for discussion if there is any, right? So the last thing that I wanted to touch upon is the, mostly on the drug development, right? So what do you mean by drug development or drug discovery process, right? So once you have a disease, what we want to have is a drug that can be consumed to treat the disease. Right, so let us say today I have fever, I just take 650 milligrams of paracetamol, right? But then how did we get that one? So that goes through a very, very long, stringent, and very, very expensive process before we could come up with a drug that can be used for treating a certain disease, right? But then in the last less than 5 years, right? So what we have seen is that a large number of data-driven methods, right, have been used in every step along this pipeline, right? So, but traditionally, if you look at it, right, going from here to here, it's mostly the forte of biologists, virologists, epidemiologists, and so on. That's who are working in this area. So now if you go from here to here, it's mostly the medicinal chemists, organic chemists, pharmacologists, and things like that. And then when you go from here to here, it's the physicians and the clinicians come into picture. And of course, a large number of different expertises are needed for you to go from here to left to the right. But one thing that is common that we have seen in the last 5 years is that in every single step, right? So AI methods has been making a huge impact, or we see a large potential for these methods. Right, I'll just give you a indicative number. Right, in the beginning of 2018, right, which is not too far from now. That's only 4 years before, right? So the number of startups, the tech, the deep tech startups. I'm not talking about the fundamental sciences. The deep tech AI-based startups that were there that were started until at that point for AI for drug development were about 80 or so in 2018, right? In less than 1.5 years, that number grew up to almost 300. Right now, after 2 more years, now we don't even have a count of these things. That's the number of companies, tech companies, right? Recently, some of you may have heard that this company owned by Google, it's polymorphic, I think it's polymorphic. It's a new company they started called Polymorphic, which is Google's company for AI for drug development, right? So, you see large number of, companies coming into this particular area, right? So one of my own, students is now working with Facebook AI, where, there's nothing to do with Facebook social media, but, he's in a team which works on AI for molecules. Okay, so in the last 3 years or so, we have been contributing heavily on some of these aspects of modern machine learning based methods for molecular design, right? So I will maybe take a couple minutes and then explain one of the projects, and if there is time, I will talk about one more, right? So what do we mean by identifying a molecule, right? So if we take drug molecules, right, let's say paracetamol. Paracetamol is a small molecule, organic molecule, which binds to certain proteins in the body, which helps in treating, in reducing the body temperature or in for relieving of the pain. Okay, so every drug has its own mechanism by by which it works, right? But then let us say we have COVID-19 now. How easy or difficult is it to find a molecule that can be used for treating COVID-19, right? So basically, and maybe I will go to this one. You see the blue, large blue one, right? So the number of, just in theoretically speaking, the number of such molecules that are possible are in the order of 10 power 60. So what is the question that we're asking? Is it possible to find one or few of these 10 power 60 number of molecules that can be used for treating COVID-19? That's the question that we're asking, right? Of course, it's just, impossible, right? You can't think of swifting through all these theoretically possible molecules and then take one by one and then do a testing and then find a drug molecule, right? So that's not possible. So hence you have to have technologies and methods that can help you identify those things without having to go through all of them, right? So in that, with that context, I will quickly try to explain. So typically what is done, you take a large database of molecules, usually in the order of hundreds of millions, okay? And then you want to identify some top molecules based on, let us say, whether this molecule will help me, whether it will help me treat COVID-19 or not. Let us say that's the objective, right? So, I will not go into much details of what, the other smaller questions are. Let us say the larger question is, I have a large library of molecules and I want to identify a few of them which can be used for treating COVID-19, right? So as I said, if you have to go through each one of them, that's not going to be easy, right? So one of the things that we did, about 1.5 years ago was to use, modern ML to be able to identify this without having to go through all of them, right? So, it's like a typical, ML kind of framework where, you represent the molecules in a certain way that can be used for machine learning. In this case, we use autoencoders, and then we represent the molecules in the, in the latent space. And, based on that, we choose molecules for, doing certain evaluation, right? Whether, it is useful or not on the COVID-19 main protease protein, which can be used for treating COVID-19. And then we use, Gaussian process and the Bayesian optimization, and then based on which we do, right? So just to give some, results. So if I were to go through each one of these molecules one by one and then say whether it's useful or not, right? So this is my curve, okay? So this will approximately linearly go and then meet one at 100 here, right? That's at 100%, I will reach one, right? But that's where I want to reach. But then I cannot move beyond a certain point of my own constraints, right? It could be time, it could be budget, it could be any of those constraints, but I will not be able to move any further, right? We developed this Bayesian optimization-based method by which we showed that it is actually possible to reach almost, 1 by sampling only about 6%. We don't have to go all the way up to 100%, but just go up to 6%, and then you will be, as successful as, reaching this to 100 traditionally. All right. So, so just to give some graphic, this is a protein from the SARS coronavirus 2. And, these are, for example, that's a molecule which binds to this and possibly be helpful in treating the disease. Okay, so I think I have 5 more minutes at least. I will skip this, some of these, and then maybe I show this one, right? So this could be applicable for any given situation. So let us say my objective is to realize molecules of some use, whether it's drugs or new materials or new molecules that are used in the fancy LED screens or superconductors or any of those things, right? So if I want to achieve, or if I want to come up with these new entities that are useful, right? So how can possibly AI affect this whole pipeline, right? So first step would be, is it, right, can it actually, tell without much of experimentation, saying these possibly are the molecules of interest, right? And then once it proposes those molecules, the next question is, how do I even make it, right? At the end, if it has to be useful, it has to be made, right? So how do I make it? So can I have an AI system which will tell me exactly how to make it? Like, it will tell that, okay, I tell the system that, okay, this is the molecule I want to make. The system tells me that, okay, from this company, you get this reagent. From this company, you order these reagents and get this particular medium and so on. And then say, mix in this particular, order. And these are the conditions you maintain, and you will get that molecule, right? So can an AI system suggest me possible routes? So once these routes come out of this particular system, can AI enable robotic synthesis read this and then take the chemicals that I order from these companies and then do the synthesis by itself? And finally, there is another AI system which will confirm that the synthesis has gone successfully, right? So it could, even not even 5 years or 10 years ago, we are able to think of such a, or imagine such a pipeline, right? Where the complete, a process of going from design to realization could be automated, right? Of course, today it is not done, but then there is a lot of work that is going in area. For example, the video that you see on the screen, this is an actual system which does chemical synthesis, right? Driven by AI, the robots, do this, synthesis. There's, again, companies like IBM and several other startups have been successfully create such systems, right? So I will go to one example and then I will close, right? So let us say, don't worry about what is on the slide right now, and then maybe I'll try to make it simpler, right? So let's talk about a drug molecule or a tablet, right? So let us say, taking tablets are pretty common, right? So let us say I take a tablet. What happens to it, right? So what happens is, we introduce the tablet to the digestive system, and these molecules that are there in the tablet has to get into the bloodstream. It has to, be soluble in the blood. It has to go to the heart, and then the heart can pump the blood with the drug molecule to all the parts of the body, and hence it can work. And after it works, it has to come back to the liver, get metabolized, and then the metabolized drug has to be excreted, whether in urine or sweat or feces, right? It has to be excreted, right? So that's what happens. But then all molecules cannot do it. You could have a magic drug which solves your problem, but then if it is not able to, get absorbed into the bloodstream, or if it does not enter the cell, right? Or if it cannot be excreted, right? This molecule is useless, right? So that's what you see on the right-hand side top, right? So let us say I have a molecule which is given in blue, right? So once these molecules are there, if it has to enter the cell, so let us say this is the inside of the cell and that's the outside of the cell, right? It has to go from outside of the cell and go inside, right? So how do I know whether it goes inside or not? Let us say this is log P is some quantity which will help me decide whether it will go reach the inside of the cell or not, right? So that one is good enough for us to understand what it is that we do, right? So let us take this particular graph, okay? So what we're saying is, like, this is the value that helps me quantify whether this molecule can enter a cell or not, right? So if the value is between these two blue lines, then they can enter. If it is below that or above that, then they cannot enter. So this particular molecule is absolutely I shouldn't say absolutely useless, but not very desirable, right? Because it has to reach the cell for it to work. So now we used reinforcement learning to see if we can generate molecules that are only in this particular region, right? If I do a normal generation and do a distribution, this is how the distribution looks like, right? These are regions which I'm not interested in, right? Is it possible to generate all of them within this region, right? So that's what happens. If I use reinforcement learning, you'll see that the distribution is slowly moving towards the region between the two blue lines. And most of the molecules that are being generated has properties that are of my interest. So similarly, let us say some other metric which says that I don't want to be here. This would be the most comfortable situation for me, right? So normally, without any conditioning, this is my distribution. But then if I use reinforcement algorithm to do the generation based on certain reward functions, I can push this distribution to the left where we want it to be, right? Okay, so similarly, we use the same kind of algorithm using reinforcement learning to generate new molecules that can strongly bind to this COVID-19 main protease protein, right? Okay, so with that, I will quickly conclude. What I was trying to do was trying to give a larger picture of how AI is impacting healthcare, and then I took different areas. One is in diagnosis, second in prognosis, third one is in the outbreak prediction and disease surveillance, and then these possibly are the ones which will help us taking healthcare to all. And finally, the last example I showed is the AI can actually play a big role in making drug discovery faster and less expensive. So now you can ask a question saying, okay, 2 years down the line, there are so many algorithms, so many methods, so many news about how AI is doing. Where is the application, right? So I said, we could, do a risk stratification, right? So, but why can't we do it now? Or why couldn't we do it during the second wave when people were, a large number of people, died during the pandemic, right? So the question, that's a, question that has a lot of, it is possible, but then there are several other limitations in terms of why we are not getting there, right? They can be easily addressed when people come together. So one example where, AI, was showed very well in advance. Actually, this was in July 2020, if I remember right, or end of 2020, where when Greece decided to reopen its borders during the pandemic because the economy was failing and they had to have tourists coming for their economy or this country to survive. So they said, we are going to open up. And then they started working with people and then see if they could, identify people who are possibly infected and then minimize the number of tests. And hence they could actually, use less number of test kits, because in the early pandemic, even if you want to get an RT-PCR done, it was not so easy, right? Because it took time. Now it's easy. But at that time, they could, use the reinforcement learning to allow, people, when people are coming from outside, right, the country, visiting the country, right? So their details go into this AI system, right? And that AI system decides whether this particular passenger has to be tested for COVID-19 or not, right? So if the algorithm says this patient is possibly not at, the risk of infection, they can be allowed, to go out of the airport, but then, there are people who are identified who are most likely infected, and then they are tested, and then you continuously inform this. Then this was a huge, reasonably large success of how AI, again, this is one example, there are several other examples, right? So with that, I would conclude. And then of course there are larger questions like ethics and the data privacy, right? If I go to a hospital and then get some treatment and get some diagnostics, right, the data belongs to me, right? As a patient, the data belongs to me, right? The hospital cannot say, okay, that I have a friend who is doing research in AI, can I give this data to the the person who is working on AI? No, the hospital has to take my consent before my data can be given to somebody, right? So there are several other large things that are involved in, like many other areas, how the careful one has to be handling data, algorithms, implementations, and so on, right? So finally, I just wanted to do a little bit of a campaign. And Professor Bapi Raju mentioned about the Technology Innovation Hub, which is the iHub data at IIIT Hyderabad, right? So we are, reasonably large entity where we undertake research and development and startups and translation research and so on in different areas. One is in data engineering, AI for healthcare, AI for smart mobility, and then whatever comes up with do productization, translation, and then help people in doing startups and encourage entrepreneurships and so on. So those who are finishing their BTech or MTech or doing PhD or working somewhere, people at all levels, we are expanding our team to have more people to possibly make an impact in our society, especially. So please let us know if you're interested. So with that, I will stop. Thank you very much for your kind attention. Thank you.
Thank you, sir, for such an insightful presentation on role of AI on COVID-19. Now I request the Bapi sir to please initiate the discussion.
Yeah, any questions from participants? Can either raise your hand or unmute yourself.
Bapi, can I ask a query?
Yeah, go ahead. Yeah.
Sure. Thank you, Professor Priyakumar. I hope when I addressing, I took your name correctly. So thank you for a nice presentation and enlightening on AI and its use for COVID pandemic. In fact, I will have a couple of a light comment, remark, and also two queries. One is that AI is as infectious as COVID-19. Today we see that everybody talks about AI in every domain, and it is as infectious as COVID-19. So that's a remark. It's a remark on AI actually. So I know that AI has already a one death because of overstating itself in 70s. So I always tell that the AI must have something should tell the no, really not really over market it itself. So I'm really tempted to add one thing is that my son has recently edited and Dr. Bapi knows my son, and he has edited an issue on AI in life science. I will send it to you. I used to discuss with him. One thing is that the real criticism on AI system is the power of explaining. Which the Feigenbaum of the first avatar of AI in expert system, the explainability was its strength. Yeah. But particularly now today, connectionist model does not have that explanation. So, what is your remark on that? What is I do not know the more research in this area, but I am just my general query.
I think you're absolutely right, and then hit right on the point. So, of course, large number of these, the solutions that are being come up with, these are a lot of them are just black boxes, right? Though they promise high accuracy and so on, we do not know how it works, right? But then, the recent focus, right, has been on explainability. So we ourselves have done some work. So the first one that I mentioned about the ECG classification, where Professor Bapi Raju led that project, there, though we classified ECG signals for certain conditions, right? We also try to identify or learn how, or where is the attention on this ECG for identifying that particular condition. And then based on those attention maps, we actually went back to physicians and cardiologists and then checked with them, saying, is this, if this is the ECG signal, and if this has this particular outcome or this particular condition, right? So is this the region based on which this is being implied or diagnosed, right? So there are several research studies that are going on to increase the explainability, and this is especially important in healthcare, right? So when we go to physicians and say, oh, this works, the first thing they ask is, please explain how it works, and hence people go back and then try random forest like approaches, decision tree like approaches, and then try to come up with an understanding. But you're absolutely right. There's a lot more research and a lot more efforts needed to make these systems more explainable and interpretable. Yeah, absolutely.
So the second query is that you have, of course, in your last slide you have put the word ethics and trust and all this. So what I am really little bit worried as well as curious to hear from you. Once you told that the probability of survivability. So if if suppose like a patient is told to his family member to say that the survivability is 60, 70 or 50%, so the point of ethics comes that should we abandon the treatment or no? So what exactly, because the corporate hospitals are very expensive. I joke with my kids that if this is what the limit, if you see that the treatment is going beyond this, then you leave it, let me die, so I think
that is again a very important pertinent point, actually. Yeah, I mean, but the like many other diseases, right? I mean, the whole idea is to basically risk profile, because we know, even in the first wave or second wave, most of the population recovered without any problem, right? But then if we knew who are these people who are at high risk 10 days in advance, right? Maybe we can care for them better, right? And increase their, the possibility that they would survive, right? That's the whole idea. I don't think this is a system that should be an app or something like mobile app where, I enter some numbers and then I decide whether I will survive or not.
I think to add to your logic of line of thinking, I think the more money can be channelized instead of
absolutely
putting the money for the blindly for others.
Absolutely.
The good cases, maybe
yes.
The difficult cases.
Yes, yeah. Money is one thing. Second one is the available resources, right? I mean, there were situations where doctors were not available, oxygen was not available, beds were not available. So, optimizing the resources.
Optimizing the resources. Yeah, that's
good morning, sir.
On a lighter note, I think if you give your personal threshold, 55 lakhs is my threshold. AI can optimize, your chances and give you now you should get out, pull the plug.
So in fact, even that 55 lakhs for a given time period, AI can help in invest it and get some money, earn some money in that time period also. Put it in money in the Okay, maybe Dr. Lipika is asking something.
Yes.
Yeah.
Yes, sir. Thank you, sir. Good morning, everyone. Sir, my question is a bit practical related. Sir, actually, I'm right now working in diagnosing DR, that is diabetic retinopathy. So, sir, my question is using Bayesian optimization, is there any other approach for hyperparameter tuning when we deal with this type of pandemic diseases?
I mean, at some level, right? So when you are trying to do a classification or a regression, at some level, it's a machine learning problem, right? Of course,
Yes,
the inputs or the embeddings or the features that you derive and then use it for your network to predict. That comes from an expert, and that is absolutely necessary, and it's very, very important for the model to perform good. But at some level, it doesn't matter whether this is for your for pandemic or for image classification or for predicting stocks. It could, at the end of the day, they are all similar in terms of optimizing the or training the network itself, right? So there's nothing special. Of course, all the care that one has to take to avoid overfitting and for avoid data leakage and all that we have to ensure. But at some level, this is a machine learning problem beyond once you have featurized the input, right? So that way, no special thing for pandemic. Yeah.
Yes, sir. Thank you, sir.
Thank you, sir. We also have Sarup, sir. Yes, sir. You could go ahead.
Professor Priyakumar, first of all, excellent presentation.
Thank you.
And I loved it really.
Thank you.
So, very, a lot of information and a lot of perspectives. Excellent. I had two questions. One is in the last you mentioned the use of reinforcement learning for design of molecules. And I want to know what are the kind of actions that you have in the model, and did you have to use any kind of simulator to predict the action? The output states, the next states, or did you obtain this from some kind of observations?
Yeah. Okay. So good question. So basically, the action is this: we have a pre-trained generator model, right? So we have large databases of molecules available. Whether they are not, whether they are useful for a certain for a given purpose or not doesn't matter, right? We know what a molecule is, and then we know the language in which the molecule is written. Let us say I have a one-dimensional string that is unique to a given molecule, right? So I have millions of them. So based on that, we have a RNN-based network which speaks the molecular language, right? So they can generate valid molecules. So that's one. So once this is my action, and then once these molecules are generated by this RNN, now I can do an evaluation, or basically, I want to know, or what are the properties of this particular method. So in this case, we could use some simulation, or we could use some neural network based method and then calculate the property of the molecule. So now, what is the objective though? Objective is, like in this case, I want molecules that bind to this SARS-CoV-2 main protease stronger, right? So I want the number that comes out of this particular simulation to be a larger negative value. So now the reward function is like a exponential function where larger negative the value of the property here is, the reward will be higher, right? So, and that's the the actor critic network based on the value from here, and that based on that policy gradient, we they optimize the generator. So that's the the generator is rewarded if the molecules are generated or have a higher affinity. It will be penalized if they have a lower affinity. So, and hence we generate and then show that we can actually move the distributions with respect to the desirable properties. I hope I answered the question.
Yeah, I will also refer to the paper if it's published. The second question quickly was actually about trustworthiness. A lot of questions were already asked by Professor Mahanti. One particular thing that I wanted to kind of point is about the robustness of these models. At least we have worked on non-image based models, neural networks and all. And recently, formal verification is being used. It's becoming a fast-growing field for ensuring the robustness of neural network-based models for range analysis, etc., or misclassification and those kind of things, so that it will ensure certain properties that you know should hold. And there are techniques using where I think DeepMind works are there. They include symbolic knowledge during the training of the network itself, so that your constraints, symbolic constraints, etc., will kind of guide the weight generation process, essentially. Process.
Yeah,
So
I mean, we go
that
through
is another
different.
field that,
Yeah, sorry, please go ahead. Yeah, sorry.
Yeah, that is one of the things that we are actually trying to see. This ultimately, these models are programs, and how do you answer the range? How do you test them, for example?
Yeah, absolutely. You're right. So, we do different, especially in the context of molecular generation, we, evaluate them based on different parameters, based on the validity of the model. And then the second one is novelty. When it does a generation, how different or how similar is it from the training set, and within the generated, molecules, how diverse are they, and different kinds of metrics. And then of course, there's always room for improvement. That's, yeah, thank you. Thanks.
Thanks.
Thank you, sir. With this, we come towards the end of the session. Thank you, Professor Deva, sir. Thank you, Bapti, sir. Thank you, Shikesh, sir, and all the faculty members and all the participants. With this, I, Ritika, signing off from the first session of 18th ICDCIT 2022. Thank you, everyone. Have a great day.
Thank you.
Thank you, sir.
Thank you all, Dr. Swaroop and Dr. Satya, Dr. Shandip, Dr. Bapiraju. Thank you very much. See you then. See you in the other session.
Thank you.
Thank you, sir. Bye.